Good evening, everybody. Welcome to BrisScience for December 2019. I am your host for this evening, Joel Gilmore. And of course, BrisScience is brought to you by the University of Queensland, where we aim to bring not just the best scientists, but also the best speakers to share their knowledge and passion for science. We're hosted here at The Edge at the State Library of Queensland, our fantastic venue partners. And of course, I would like to acknowledge the traditional owners of the land on which we're meeting tonight and pay my respect to elders, both past and present. Tonight, we have a very special returning speaker who I'll introduce in a moment. A couple of bits of housekeeping first. If this is your first BrisScience, thank you so much for coming along. We do a couple of things a little bit differently here at BrisScience. Firstly, because this is more of a social event than a lecture, particularly at Christmas time, we're going to be having some food and drink afterwards, so we'd love for you to hang around and join us and have a chat. We also take questions a little bit differently. We take questions either over Twitter with the hashtag BrisScience, or you might have picked up one of those question slips on the way in and you can write your question down and we'll get to as many of those as we can. Or feel free to put it on a Christmas card or anything else festive you might have. And we would, of course, ask that you put your phones on silent, but feel free to tweet throughout the night, as we will be doing live tweeting through the evening, taking down some of the highlights. So you can catch up with that later. And this and all our BrisScience lectures do get videoed and will go up on our website and onto YouTube, so you can catch up on them again later and see some of those highlights. So tonight, as I said, we are having a returning speaker, which is Associate Professor Bryan Fry from the University of Queensland's School of Biological Sciences, where he runs the Venom Evolution Lab. And he is a world expert on venom, everything from toxic snakes to other equally terrifying venomous creatures. He has led expeditions to 40+ countries around the world, including Antarctica, which led him to be inducted into the elite professional adventure society called the Explorers Club. And tonight, he is going to share some of his latest research on snake bites and treating snake bites, particularly alternative treatments for remote communities. I do, however, have to point out that snake bites are a somewhat terrifying business, and so apparently some of the images tonight are very detailed of those bites. I'm not saying he has made a child cry before through a talk, but, you know, if you are easily terrified by graphic images, you know, just sort of cover your eyes for some or all of tonight's talk. I'm sure you'll be fine because we're all scientists here, or enthusiastic or grossed out, whatever it might be. But for now, for a bite-sized chunk of snake science. Thank you. I've used up all of my puns on the previous week's privilege. Please welcome Associate Professor Bryan Fry. Thank you.
All right, well, thank you very much for coming. And yes, some of the slides are a bit gnarly, so I hope you enjoy them. All right, so what are snake proteins, first off? Where does venom come from? You know, it's not like there's an intelligent design fairy who's a bit hungover and decides and say, here, have another toxin. No, it doesn't work that way. So all venomous animals are using modified parts of their body as a weapon. So in the case of tiger snakes in Australia that cause you to bleed out of areas you don't want to bleed out of, their venom is loaded with an activated form of a clotting enzyme, so something that we would use in our body to make a blood clot. Or in the case of a death adder that causes you to have a nice little limp paralysis, they're using a modified brain neurotransmitter to bind to your nerves and prevent signals from getting through. So you can think of some of the venoms as being overdoses, such as the tiger snake, where the venom effect is exactly the same that would happen if I took recombinant human factor 10A and gave you an injection of it. That's basically what's happening with a tiger snake. Or something like the death adder, where it's a dramatic underdose, you might say, you know, where it's cutting the supply line. So in this case, none of your nerve signals are getting through, telling your muscles to contract, which means your diaphragm doesn't move, your lungs don't move, you don't get any air exchange, you don't get any oxygen, and you die. All right, so a little bit of housekeeping. Venomous and poisonous are not interchangeable terms. So snakes are venomous, but something like a fugu or some of the really toxic clams, those are poisonous. So with a venomous component, it's something very, very large, such as this protein here, where something like ciguatera toxin, which a major food poisoning component in seafood, is very small and would fit into the corner of the snake toxin. So it's a case that size does matter. So this means that you can drink venom and you won't have a toxic effect because the components are too large. They need to create a wound to get it in you, versus something like that's poisonous, very, very small toxins that can absorb through your skin or create intoxication when you eat them. So what are venom systems? So these are key evolutionary innovations that have evolved for three main purposes that are not necessarily mutually exclusive. So you have it for defense, such as stingrays. You have it for prey capture, such as a snake eating a mouse. Or you have it for competitor deterrence, such as the Australian platypus, which is also a great example of why the female of any species lives longer. So in this case, males are stabbing each other with intensely painful venom to determine who gets the breeding rights. Yeah. We haven't gone very far from this as humans. If you disagree, what are some of the famous last words of the human male? Hold my beer. So showing off is something that is, the prerogative of the male of any species, which is why living longer is the prerogative of the females of the species. Venoms have evolved on at least 25 separate occasions throughout evolution. So it's one of these ones where it's a key evolutionary innovation where there's more times that venom has evolved than flight has evolved. So it's something that we see over and over and over again. And you can see for a number of different functions all over the tree of life. So we see it across the invertebrates, where it's evolved on at least 6 separate occasions, such as our centipedes, bees, wasps, hornets, scorpions, pseudoscorpions, spiders, and there's even lethal caterpillars. Death by caterpillar is probably the least glorious thing to have on your tombstone, but to put this in perspective, 50,000 people a year in Brazil alone require antivenom for caterpillars, where there's lethal caterpillars down there, a lot of which are very brightly colored. Let me just go back. Usually to deter the smart primates, which I wasn't. My very first envenomation was from a brightly colored caterpillar. Anytime you have a slow-moving, brightly colored object, if you have half a brain, you know not to pick it up. Clearly, I had less than a half a brain. So it's one of these cases of where if you see a slow-moving, brightly colored object, you're an animal, don't pick it up. Now, we have across the cnidarians, which is the oldest venom system at over 700 million years. But even more impressively is if you think about it from the perspective of generational time, where your jellyfish can hit sexual maturity in as few as a few months. So that means that compared to something like a king cobra, which might take 8 to 10 years to hit maturity, that you could have 40 times more generations of jellyfish in that same period of time, however many tens of thousands, hundreds of thousands, or millions of years. So that means that the diversification, because every time you have sexual reproduction, you get some errors introduced, and that's, of course, the basic substrate for any evolution. So the diversification of the cnidarians is astounding. It's the oldest and most dynamic venom system, but it's one that we know the least about. We thought we were really clever a few years ago when we realized that more papers are published in 1 year on snake venoms than have ever been published on jellyfish. So we thought we had discovered this hidden area of venom, and then we realized why. After it took a really, really OCD PhD student to grind through this miserable project and get a degree out, and we haven't touched jellyfish since because they're horrible. It's basically really, really unstable snot is the best description of jellyfish venom. By the time you get the snot component removed, the whole thing promptly falls apart. It is the worst venom system. And of course, we didn't publish that as a cautionary tale. We probably should have. So there's probably other people's hopes and dreams being wrecked while they think that they've discovered this great untouched resource. Anyway, so across our gastropods, we have venom evolving in the last common ancestor of our cuttlefish, ockies, and squid, where the blue-ringed octopus isn't the only venomous lineage. If you go diving anywhere across the world, you'll often see clamshells or other bivalves that are opened and they've got a hole in the side. And that means that an ockie has drilled through the side and injected their venom, causing paralysis and relaxing those muscles. So all of the ockies are venomous, but only very few of them are dangerous to humans. And that's one of the reasons why so many different venom systems have gone understudied, is that there's been a very strong human bias. Of course, there is reasons to study the ones that are important from human medical perspective, as I'll get into later in the talk. But this being Australia, of course, everything wants to kill you, including even the seashells. So we have the cone shells. There's other types of venomous snails. There's even venomous worms. There's, of course, the sea urchins, which I'm convinced that the road to hell is paved with. I've been stung countless times by these wandering around the beach looking for other animals and having to get out the razor and do a bit of bush surgery. Yeah, good times. So within the echinoderms, venom has evolved on two separate occasions, one of which is the crown of thorns, a starfish that gets a really bad rap. It's a really, really cool animal, and it's our fault that it's flourishing across the Great Barrier Reef. It's a native animal. It's natural to the Great Barrier Reef, but the reason that it's flourishing and destroying so much of the coral is several-fold. First off, we've hunted into virtual extinction the trochus shell, which is the main predator of the crown of thorns. So that means a lot more baby crown of thorns are living to become adults. Secondly, their main prey, which is coral, are very, very stressed out because of all the agricultural runoff that's creating algal blooms and sediment that is smothering the coral. So they're immunocompromised and they're not able to defend themselves as well against the crown of thorns. And of course, the corals are also stressed because of water temperature. So that means that the crown of thorn starfish is blooming into these plague numbers, but it's our fault. Like I said, they're a really cool starfish. Then we have the other major venom system within the echinoderms are your sea urchins. So your typical urchin here, long spines, like really hurt to dig out with a razor. There's one really cool type, the flower urchin, which actually has venomous claws. They're pretty spectacular. I haven't been done by them, one of the few ones I haven't been done by, but I did get to watch a friend of mine get done by it. And it was, classic case of, well, why men don't live as long. His protective gloves are on the bench next to him, and so the story starts there. And then, for reasons that remain unknown to this day, he decided that it'd be a good idea to move a slippery circular object from one tank to another using chopsticks. Yeah, this is going to end well. One thing about people who work with animals, cause and effect, they have very, very good reflexes. So as the as the flower urchin predictably slipped out, he caught it without thinking and promptly got envenomated really, really badly. He said beer helped with the envenomation. But he's got a pretty good sense of humor about these sorts of things. He's working on describing a new species of irukandji jellyfish and naming it after his ex-wife. Describing the envenomation as a short but painful experience. So, across the vertebrates, getting closer to home about the talk, we have, with the reptiles, that venom evolved in the snakes very early in their evolution, using the same starting substrate that the venomous lizards did, but they're not the only of the vertebrates. Like, we just had a paper come out in Proceedings of the National Academy of Science on the Solenodon, which was written up about New York Times and other places. That came out a fortnight ago. But that's one of several venomous mammals. That within our insectivorous mammals, venom has evolved up to 6 times just within that lineage alone, which includes the water shrews. You have the platypus from the painful venom mentioned. And then we're even working with a venomous primate called the slow loris, which is an animal that uses its venom for defense. And anytime you have a defensive venom, it's very, very painful. When I was living in Singapore and doing most of our work that we've been publishing on that, I did in Singapore while I was working at the National University of Singapore. We had a big collection of slow lorises. We had a visiting vet student at the zoo where we were keeping the animals. And like most new medical graduates, the Dunning-Kruger effect was strong in her. So she knew everything even though she'd only worked with green macaques. So she sees me putting on these heavy leather gloves with metal bits embedded into them, looking like I'm about to go jousting in Game of Thrones or something. She's like, no, I can get it, watch. Sure, let me just get out the camera first. I'm a big believer in letting people control their own destiny as long as I can laugh about it later. So she gets it perfectly behind the waist and behind the back of the neck here, which would be great if it was a macaque. Now, had she let me finish what I was going to say to her, was that slow lorises are one of the earliest splits off the primate lineage. They actually split before the ability to move the eyes left or right evolved within primates. So to move their head, or to look left or right, they turn their entire head, giving them an owl-like flexibility to their neck. So the head spins 180 degrees like something out of The Exorcist, and chomp. Yeah. So within our fish, we have venom evolving on two main occasions, in the sense that countless times within the fish, but of all of weaponizing the big dorsal spine. So a stingray has weaponized the same spine as a chimera, as a Port Jackson shark, as a stonefish. They've done it independently, but it's doing the same thing over and over. In comparison, we have the fang blennies, or vampire blennies, the coolest little fish. They use their venom for competitor deterrence, where they're fighting over who gets that perfect little piece of coral to hang around. So if another fish comes by and tries to take their little piece of coral hiding spot, they bite them rather than stinging them with a dorsal spine, but they inject them with these opioid peptides. So they get their competitor really, really stoned, which means that they're now happily baked and swimming out in the water column and getting eaten by something larger. Problem solved. So it's a pretty ingenious way to get rid of your competition, is to remove all of their survival instinct. All right, now on to today's talk. So snakes, they use their venom for predation. Some of them will use it for defense, as we'll see, but snakes do not attack. Snakes only defend. Another way of thinking about it is snakes don't start fights. They end them. So in this case, whenever a snake bites a person, it's because they feel scared. They feel threatened. If you have enough time to go into your garage and get a shovel, clearly you're not in immediate danger. You should be calling your Parks and Wildlife and having a snake removalist come out and safely do it. Snakes are a protected animal. It's no different than tearing the head off a kookaburra, if you kill a snake. Anyway, if you go get the shovel and you're not in immediate danger, you come out, you try to kill the snake, you only injure it, it bites you in the leg. You do not have the divine right to throw yourself to your knees, raise your arms to the heavens, and plaintively cry, "Why me?" Because you're an idiot, that's why. So with snake bites, you can kind of divide them into two different types, which I'll show in the next slide, legitimate and illegitimate bites. But how many people get bit? Well, first off, despite everyone in Australia loving the fact that we have a lot of venomous snakes, more people die from sexual misadventure each month than die from snake bite over an entire year. So with that sort of thing, to put it in perspective, about 1 to 2 people die a year in Australia. A really bad years, we might have 3. To put that in perspective, every 10 minutes, somebody dies in India from snake bite. Now, every 3 to 5 minutes, somebody's getting bitten in India. It's one of these kinds of scenarios where India alone has at least 1 million snake bites a year. We don't have a proper handle on how many, because of course, the countries that have the worst snakebite problems also have collapsing infrastructure from the burden of malaria, dengue, HIV. It's under a continual onslaught from a number of other aspects. But we know that the only thing we know is how little we know. That the upward estimates are about 5.5 million, but even those are recognized as gross underestimations of how many people, because a lot of people aren't getting bit and going to the hospital. They're seeking traditional healers and dying out in their village, or they do go to the hospital, but there's not good record keeping. So it's one of these things that, because the numbers are so poorly documented, snakebite's not getting the attention that it deserves. Where, to put it in perspective, $200 can save someone's life. $200 can prevent someone from losing their arm and the entire family being plunged into poverty. If you have something like malaria or dengue, these are much more complicated, expensive treatments. If you have something like HIV, it's obviously a much more ongoing economic drain. So no other catastrophic impact disease can you have such a tremendous benefit with so little money, yet snakebite has only just been put back onto the neglected disease register of the WHO. It's been very much neglected. So who is getting bit? So we have our legitimate and our illegitimate bites. So legitimate are people working in farms. If you're working out in the rice paddy fields, obviously you're not going to be wearing shoes because they're just going to get sucked off by the first bit of mud. Well, you're also going to step on a Russell's viper. Something like sanitation, the lack of indoor sanitation in India, for example, is one of the major contributing snakebite variables for snakebites to women, because they're holding it all day long. They don't want to go out and use outdoor toilets during the daytime. So they hold it, they go and they use the toilet outside, open-air toilets at night, when they're much more likely to step on a snake. And then, of course, children at play. But in the developed world, it's the 3 T's. Yet again, males. Testosterone, multiplied by the amount of tequila, or beverage of choice, multiplied by the number of tattoos, gives you your snakebite likelihood. Well, I've got 2 out of the 3 of those. I don't drink, but I've got a lot of testosterone and a lot of tattoos. So, as my wife always tells me, those days are behind me. Responsible family men know. So, who's biting? Why are we having some of the bites? So, habitat destruction is going to displace the animals. So if you clear a bunch of bushland, park a bunch of houses there, the snakes come out from wintering and they're going to go, oh, there's a nice new cave over here, and they go into what is now someone's garage. Different farming practices. So depending on the type of farming, you're going to create basically a snake Hilton for a particular species. So, in New Guinea, for example, with the sugarcane farming there, that's created a plague of rats. Well, taipans are a rat specialist. In India, in the slums, you get, yet again, a lot of rats. Yet we're seeing a trend here where if you create an area where there's a lot of new food, there's going to be a particular snake that benefits. So in India, if we want to catch a cobra when we do our field research in India, we could spend all day in the forest outside of Bangalore and catch 2 or 3 cobras. But if we go into the slums at night, we can get 18 to 20 cobras in just a couple hours. So these are cobras that have adopted an entirely new lifestyle. Instead of coming out during the day, they're coming out at night because that's when there's a lot of food around and there's not a lot of the predators, or what they perceive as predators in the form of people. But you now have these slum cobras are now behaviorally distinct from sort of the ones in the native environment, because they've switched their behavior. So there's no or very little gene flow, and they're actually quite isolated. And for some of the older slums like Bangalore in India, these snakes are actually really quite distinct now because they've been so isolated from a different behavior. Rice paddy fields, best thing that ever happened to Russell's vipers in India. In Nigeria, with the different type of farming practices there, it's created a Hilton for the saw-scaled vipers. Or in Australia, if we want to catch a brown snake, we just go find the first chicken farm that we can, because chickens are very, very messy animals, lots of food scattered all over the place, lots of mice, and brown snakes love mice. So we create an environment where we now have a greater density of snakes in this disturbed environment than we do in the natural world. We, of course, have poverty being a major contributor to snakebite, where, like some of the work we were doing in Pakistan about helping people snake-proof their houses and office areas, that when you have extreme poverty, not only do you have dwellings that are not exactly sealed, but you also have people sleeping on the floor. So they're much more likely to be bit by a snake in that regard. And then we have climate change, where, as we all know at the moment, things are getting hotter and it's staying hotter longer. Well, that means that the snake activity period is also persisting longer, not just through the night, where the night itself is warmer and the snakes can be active more. They might have, before it might have been 10 p.m. is too cool for them. Now all night long it could be warm enough for them to be out foraging. But remember back, mid-winter here, we hit, 34 degrees at the beginning of August. There was already snakes out. There was a snakebite death in New South Wales at the end of July, which was almost unheard of. So it's one of these kinds of things that with climate change, that's also a big contributor in changing the dynamics of snakebite. So what do you do in the case of snakebite? Well, you don't run around like an idiot, because that's just going to move the blood around. First thing to do is keep the person calm. get them down, bring help to them. Best piece of first aid, car keys. Bring help to the person, or if you're in an urban area, you can call 000. But any first aid course will teach you how to put on a pressure immobilization bandage. And that's because initially the venom isn't actually in the bloodstream. It's trapped in the fluid around your blood vessels. Like when you wipe out and skin your knee and you have that clear fluid coming out, that's your lymphatic fluid. That's the fluid, that's our second circulatory system. We have two circulatory systems. We have our blood, but then we also have our lymphatic system. So initially, venom is in the lymphatic system, doesn't enter your blood until it reaches a lymph node like in your armpit. These are the things that get all swollen up when you have an infection, like when the bulges here when you have a cold, those are lymph nodes. Or if you have an infection on your hand, you'll get a swelling here, that's your lymph node getting swollen. These are what we use to screen and purify out the lymphatic fluid, but that's also how venom will enter in. So if you gently squeeze your arm with a crepe, one of those stretchy crepe bandages, but that will stop your lymphatic fluid as long as you keep the arm also very, very still. But the key is that you don't want the bandage to be so tight that the fingers turn purple, even though that looks really cool. That's not going to help you out. And the worst thing you can do is a tourniquet, where you stop the blood flow entirely, because all you're guaranteeing at that point is that they're going to end up a cripple. So if you go out in the bush, you should know how to. You should have an up-to-date first aid certificate for more mundane things like, compound injuries, bone sticking out of the leg, those sorts of things. But they'll also teach you how to do a pressure immobilization bandage. And these can work really, really well. Like there's been cases, for example, of a person presenting about 4.5, 5 hours after a taipan bite, no symptoms at all. They got the IV in, got the antivenom ready, took off the bandage. About 10 minutes later, the venom hit them like a freight train. So it can be that efficient. It can really retard the spread of the venom very, very efficiently and save a person's life. And this is particularly important for something like a brown snake, where they're the only snake in the world that, in the absence of a pressure bandage, can kill as quickly as 15 minutes. And the thing is, it's not a steady decline. You're going to feel fine for the first you're going to feel fine for the first 13 minutes, and then you're just going to go off a cliff. Where from feeling a little crook to death with a brown snake can be just a couple minutes once they start hitting that. And there's been deaths regularly reported as quickly as 15, 20 minutes after a bite. So that's why something like a brown snake, it's very, very important to get that pressure bandage on as quick as possible. Any kind of snake bite, unless you're an expert and you know it's a python, anything else, you have to get to the hospital and keep them there for up to 24 hours. Because sometimes the venom can actually get trapped in the flesh and symptoms might not show up until 15, 18 hours later. I've seen that happen with death out of bites, where it was about 14 hours after the bite, he started feeling a bit crook and just went, but up until then, he felt fine, and the hospital almost released him. And I was actually, insisting that he stay, and sure enough, middle of the night, he went. All right, so what not to do? Well, first thing to do is don't chop your finger off, although you will end up, with a really cool X-ray. So this is a case where a guy got bit by his pet gaboon viper and just took the metal shears and chopped his finger off. So first thing to do is don't do that. Now, there's a lot of other things not to do. So the reason that a lot of these alternate therapies seem to work is that people were going to survive anyway. Basically, they survived despite these therapies. So it's a real problem in the developing world of traditional healers, where that's the first port of call, and that's just delaying things. And with snakebite, time is tissue. In the very least, all you're doing is making it more likely that you're going to lose something. If you want to see this in a very powerful way, watch a documentary done by my mate Ron Whitaker on his work with the Arula snake tribe in India called 1 Million Snake Bites. And in this particular case, the Arula snake tribe were the former snake charmers of India, which that's now illegal, which is good because they were doing things like tearing fangs out and sewing the mouth shut. But anyway, they know a lot about how to find snakes. So the Arula snake tribe has now been co-opted to where they're the snake catchers for the collection of the snakes that are used for the production of antivenom. So their entire livelihood is built around getting snakes, getting the venom to make the proper medicine to treat a snake bite. Halfway through the filming of this documentary, one of the main Arula guys, his mother, gets bitten by a saw-scaled viper while out collecting firewood. And rather than taking her to the hospital to get the antivenom administered that was made by snakes that they had caught in that exact area, you're never going to get a better match than this, they went straight to the traditional medicine. And there's this scene in there where the absolute impotent anguish on Rom's face, where he knows that this is not going to have a good outcome, but if he makes too big of a deal about it, it could shut down the entire cooperative, and more people could be affected because they wouldn't be collecting any venom for the making of antivenom. So he's very cognizant that there's a much bigger picture, and you can just see that he's trapped between these competing motivations, and he's trying to encourage them, and they're just saying, no, we're going to do it our way. Despite the treatment, she still survived, but her hand was just mangled by the time it's done. So you can see that with these people, where if it was ever going to have a cultural change, it should be very readily apparent with them, but even with them, they went the traditional medicine of, in this case, taking some plants and some cow crap and putting it on her hand, rather than going and getting the antivenom. So in the developing world, cultural aspects are a huge part of it, and that's reinforcing that snakebite is as much of a sociological condition as it is a medical condition, because you have to, of course, be respectful of their beliefs, even though they're not going to be helpful. But the worst thing you can do is alienate them, because then you're going to alienate everybody in that community. There's a much longer view that has to be taken. But closer to home, we have a lot of sort of alternate ideas in the developing world, one of which is using electricity. Now, this gained a lot of favor in the US for a while, and this was people who are being bitten by snakes like the American copperhead or American timber rattlesnake, which you're likely to survive anyway. So they somehow got this idea that using stun guns on each other would be a way of treating snakebite. Yeah, okay. Might be amusing, you're not going to die, but being, Joe Redneck in Alabama, they of course didn't appreciate the fine difference between AC and AC/DC current. AC/DC is not just a cool band, it's also the type of electricity that will nuke you. So one particularly spectacular case was the guy got bit, they came home, they cut the wires on a lamp and went. Now, Australian electricity having higher volts but lower amps, you're going to survive that. But American electricity is basically designed to kill you, where it's lower volts but a lot more amps coming through. And of course, it stopped his heart. And then another one was that they took the jumper cables, hooked it up to a guy's lip, and revved the engine. Yeah, the medical description of it was quite detailed, including mentioning that he defecated on himself before passing out. So there's also, of course, the good old one of alcohol, although that was usually probably the cause rather than the treatment. Vitamin C, for some reason, has gotten favor, even though that has absolutely no use. As I mentioned, tourniquets, bad idea. You're going to stop the blood. So, what do we actually do? So in the case of something that's a paralyzing snake bite, you can keep a person alive through mouth-to-mouth only for so long. You can keep them alive on a ventilator virtually indefinitely for something like a death adder, but for something like a death adder or a kite, you're not going to be able to keep them alive for very long. And that's because every time we take a breath, we dry our lungs out. Now, who here has ever broken ribs? Why is it always usually just me? Good, some other people. Ribs are horrible. You have to keep breathing, and even worse, you have to keep breathing very deeply with your broken ribs to keep drying your lungs out, because otherwise you get pneumonia. Because if you don't breathe deeply, you're going to have fluid buildup. So in the case of doing mouth-to-mouth to somebody, you're only going to be able to inflate their lungs so much. You can't generate enough mechanical pressure with mouth-to-mouth to dry someone's lungs out. So my mate, Joe Slawinski, who was killed by a kite on 9/11, right when the Twin Towers are being hit in Burma, he got bit by a kite. They gave him mouth-to-mouth, and he survived for 25 hours before he finally died of suffocation from fluid buildup. So yet again, that's why even if there's not an antivenom available, if the person's in a hospital and on a ventilator and it's this type of paralytic bite, you can keep them alive until the venom itself wears out. So why do we see so many different types of effects for snake venom. Because remember, we are just collateral damage. Venom is being shaped by evolution, and in this case, the major shaping pressure is what they eat. So for something like a death adder, where this is work that we recently published, my PhD student Christina here, that looking at 3 different types of snakes. So you have death adders. They're a sit-and-wait ambush predator. They're basically the venomous equivalent of a landmine. They can't afford to be fussy because they're just sitting there and waiting. They can't go, yeah, I don't like that. It might be 3 weeks before something else walks by. So they nail whatever comes by. And that means that their venom is an extremely complex venom. If you look at a death adder venom, it's one of the most complex venoms you're ever going to see because a mouse physiology is not identical to a frog, is not identical to a bird. So different toxins will be better against different prey. So they need a whole bunch of different toxins because at any given time against that one thing that they've bitten, maybe 20% of those toxins might actually work. Versus something like a sea snake, which eats just fish. And as you can see, its venom is very, very potent against fish, more potent against everything else. Versus if you look at the death adder, you can see that the range between the most and the least potent is not terribly great. As opposed to something like a king cobra, where it's much more potent against snake than it is even against a lizard. So you can have exquisite sensitivity. And what that means is that the most dangerous snakes to humans are going to be those which take a lot of mammals in their diet. So a bandy, those gorgeous little black and white ringed things that you can see in Mount Glorious quite easily, they're not very dangerous to humans because they're a specialist on other snakes. So when you have something that's a specialist, you can have their venom up to like 100 times more potent on their specialized prey item than it might be against something else like a mammal like us. So that's why, for example, a brown snake, which is a specialist on mice, is much more dangerous to us than a bandy, which is a specialist on snakes. So when we talk about which is the most toxic snake in the world, well, first off, all of those toxicity tests are meaningless because they're tested on white lab mice, which are not even necessarily physiologically the same thing as a native mouse, let alone what that snake might actually be feeding on. So if you take a bandy venom, you're going to say, oh, that's not toxic at all based on a lab mouse. But you test that on a snake prey, and it's going to be one of the most toxic venoms you'll ever see. So yet again, it comes down to that. And even translating from mouse to human is a very poor translation, as we know from drug testing, where drugs that work great on mice, great, you cured cancer in mice, but it didn't work too well on humans. Or that mouse is feeling no pain, but you're still going to feel your bones grinding against each other. So the mouse physiology is not identical to the human. So all that stuff about which is the most potent, the answer is always, what's the most dangerous snake? The one that just bit you. All right, so some snakes, however, are really, really weird. Like this is my favorite snake in the world. This is the long-glanded blue coral snake from Malaysia. Snake about that big, they can be up to about 2 meters long. What makes them really, really cool is that their venom glands can be up to about 1/3 the length of the body. And they're a snake that feeds on other snakes, including other venomous snakes. So they're feeding on potentially very dangerous prey animals that can have a high chance of retaliation. So if we look at what their venom does, it's similar to what a cone snail does, where a cone snail needs to immobilize a fish immediately. Now that fish isn't spasming by its own accord. Those are involuntary spasms by that fish. What the cone snail is doing is turning on all the fish's nerves. So if you're doing something where like you're trying to swim and all of your nerves are spasming, you're not going to go anywhere because this is an activity that requires incredible coordination. So we see the same thing with the long-glanded blue coral snake, where if we take its venom and put it into a tissue preparation where we just have a piece of tissue from a chicken, it will start spasming involuntarily because what the venom is doing is turning on all the nerves. There's no other snake that does that, but if it's trying to feed on something like a young king cobra or a kite, another venomous, dangerous snake, well, slithering, very highly coordinated activity. If everything is spasming, they're not going to go anywhere. So we see this kind of thing where when there's a high chance of prey escape or prey retaliation, this way of turning on the nerves immediately is something we've seen evolving on several occasions independently. So we have it with the coral snake. We also have it with the assassin flies, which are turning on the nerves of their flying prey that they're taking in the air. Yet again, something that has a very high chance of prey escape. We see it with mambas, which are feeding on birds. Yet again, very high chance of prey escape. But if a bird is spasming, it's not going to go anywhere. It can't fly with that loss of coordination. Because, of course, if it flies off, it's as gone as if a fish swims off relative to the cone snail, where even if it dies a bit further out, it might as well have gone through a wormhole and disappeared from the perspective of that snake. It's gone. And then, of course, we see it with the cones. So we see this, it's a cool area of convergent selection pressure of turning on the nerves. So this is like a methamphetamine overdose instead of your typical snake paralysis, which is like a barbiturate overdose. So the exact opposite that we see. This, of course, is something that's very useful because in all of these cases, it's extremely painful. It's not something we can use directly as a drug, but it teaches us more about how nerves get turned on and how pain is produced. And the more we can learn about that, the more information we have about how to design compounds to counteract that pain. Because there is no one pain out there. Pain is just a description, where there's a myriad of different ways that we can have pain produced in our body, all of which are requiring different pathways to be turned on. That's why if you ever get a sunburn, the best thing for that isn't tramadol, but ordinary aspirin, because the nerve receptors on the surface of our skin are exquisitely sensitive to aspirin. So that will be your number one remedy for sunburn, is this ordinary aspirin will give you the greatest therapeutic relief relative to something like tramadol, which is actually going more for your central nervous system type receptors. You're going to be looped, but you're still going to be in pain. All right, another example of how things can change relative to prey, the animal that we're looking at called the night adders. These are really cool African snakes where one species has developed huge venom glands relative to the others. They're both feeding on amphibians. But the one up here, the short-glanded one, they're feeding on small frogs that aren't much bigger than the width of the head of the snake. Versus the rhombic night adder are feeding on massive toads that can be up to 85% the weight of the snake itself. So they have these huge venom glands to produce proportionately more venom. Now, the venom itself is very similar, where in this case, what we're doing is we're just looking at changes over time for degrading one of our blood clotting proteins called fibrinogen. So if we start when it's normally intact, we have 3 healthy chains. We had venom, so here's our control. So then you can see very quickly the first chain on the top here gets degraded. Both of them do the same thing. Both of them are degrading fibrinogen. So they're removing this blood clotting component, and therefore their prey are dying of hemorrhagic shock. So in the case of humans, this will also affect the blood chemistry. But in this case, even though the snakes have the same basic chemistry of their venom, there's nothing different as far as what they're doing to their prey. Both of them are producing hemorrhagic shock. One just happens to have a lot more venom than the other. But even with this very similar activity between the two venoms, the antivenom only works against one of them, has no effect against the short-glanded one. In this case, luckily, it works against the one that produces a lot more venom. That's not normally the case. But in this, nevertheless, it reinforces that even just very subtle differences in the venom chemistry can have a huge impact on the clinical outcomes. Closer to home, we have taipans and brown snakes, both of which have incredible effects on the blood as adults. So in the case of taipans, both the babies and adults have identical venom. They feed on mammals. They're specialists on mammals throughout their entire life. So therefore, the antivenom works just as well on the babies as it does on the adults because they're feeding on the same prey. So you can see that trend. Versus brown snakes, the babies are tiny. I've literally had bigger intestinal parasites in the field than the size of a baby brown snake. So they're minuscule. So they're far too small to feed on even baby rodents. So from the egg, brown snakes feed initially on reptiles. So you look at their venom difference here, you can see that the babies have a very different venom than the adults. So the adults are hammering the blood, but the babies are actually hitting the nerves. Well, the antivenom is made using the venom from the adults. So this means that in this particular case, the antivenom is not likely to work against the babies because the baby venom is so starkly different than the adults. And this is an issue because we've had brown snakes only that big kill people. So even the very small babies that are neurotoxic are still capable of a lethal envenomation. So this is a problem. So it's one of the things we're doing follow-up studies on to see if maybe death adder, which is loaded with neurotoxins, might be a better therapeutic option for baby brown snakes than adults. But nevertheless, it's a case where you can see that changes in prey can have a very immediate impact on the therapeutic side of it for humans. So it's the perfect intersection between evolutionary biology and clinical medicine. Also, why we need to just fund basic science, because it might save your life. All right, spitting cobras. They're one of the few snakes in the world that use their venom for defensive purposes as well as predatory purposes, with the effect of turning your eye into a traffic light. But some of them also have very, very different effects. The African spitting cobras, unlike the Asian ones, are notorious for producing extraordinary local tissue damage when they bite a person. It's as if acid has been injected into their venom. Survivors often have severe permanent disability, including amputation. So we wanted to find out why. What is so different about the African venom relative to the Asian spitting cobras? Because within cobras, the ability to spit their venom has evolved on 3 separate occasions. So it becomes a perfect evolutionary model for how does the venom change relative to spitting, and therefore what is going on with the Africans. So we looked at their ability to kill cells. So the spitting behavior produces pain by killing your cells. Every time one of your cells dies, it puts out a tiny little scream that you interpret as pain. So with this tree, we have the 2 different lineages that are hooders. So your king cobras make hoods, your true cobras make hoods, but they're not related to each other. So the ability to hood up and look all big and tough has evolved twice. Now, of course, if you're just making yourself more conspicuous, you're just more likely to get eaten. So anytime, like the caterpillar that stung me as a child, anytime an animal is very conspicuous and advertising its presence, you can assume they've got some sort of chemical Krav Maga to back it up. Well, they do. So in the case of hooding, every time with the animals that have evolved hoods, they evolved these cytotoxic venoms that produce pain. But the key here is that if we look at our African spitting cobras, they're not uniquely cytotoxic relative to some of our other cobras. So why? What's going on there? Well, if we look at it a little bit closer, we start seeing a lot more subtlety. So remember those brightly colored caterpillars that were advertising their presence? Well, the same thing's happening in the cobras, that the more conspicuous the cobra is, the more painful it is, and the more likely it is to produce those severe wounds. So our primitive type cobras, they have a little bit of patterning on the front, kind of advertising their presence when they hood up, but nothing on their back, as compared to like our classic Indian cobra, extraordinarily conspicuous. Well, every time that we see these color or pattern changes to make the cobras more conspicuous, we see an increase in these pain-inducing toxins. So in our primitive cobra, they have these pain-inducing toxins relative to their ancestors that aren't hooders, so we already have a jump just with the hooding, but every time we get a color change, so we have red evolving within the true cobras, but there's also one population, the king cobra, the Malaysian one, that also is red, evolved that independently. It's the only of the king cobras that has that. It's also a lot more painful than any of our other cobras. Similarly, banding on 4 separate occasions within the cobras, these really conspicuous bumblebee-like bands evolved. And yet again, each time we have that, we see an increase in these pain-inducing toxins. So it's a great example of biological honesty, where the brightly colored conspicuous animal is going to hurt you. Something we see over and over. But of course, reversals are also teaching us as much. So twice within these brightly colored, banded, or conspicuously marked lineages, on 2 separate occasions, we had snakes that reverted back to that primitive camouflage brown. And in both of these cases, they independently had a dramatic reduction in their amount of pain-inducing toxins. So yet again, that provides more evidence for this theory linking color and pattern. But that still doesn't explain the African cobras. Why them? So across our cobras, we see all this color pattern linking Great, we know that African cobras are like that, but no more so than any of the other cobras. So it still doesn't explain what's going on. Well, one of the things that got us thinking was bleeding. That they bleed more than other cobras. So we had to look at what they do to the blood, and sure enough, there's the answer to our mystery, that the African spitting cobras are the only ones that are strongly anticoagulant, producing bleeding, but with this bleeding also helping spread the toxins, but just as importantly, they're damaging the vascular bed. So the antivenom isn't reaching it because the antivenom requires to go through the blood flow. So you have this area where local bleeding, the toxin spreading more, and then no more blood is getting through because everything is destroyed in that area. So it's worsening the venom effects and producing an area where the antivenom couldn't reach it. It turned out there's an issue. The antivenom doesn't work at all. So the antivenom is selected for its ability to neutralize that lethal paralysis, that you require to be on a respirator on and otherwise. So the antivenom isn't having as its therapeutic target the bleeding side of it. So when we tested the antivenom, that's the blue line that's hiding behind the red line, no effect. But we tested a small molecule drug that's cheap, readily available, stable at room temperature, everything you want in a drug, called vertebradib, and it absolutely hammers the venom, neutralizes this anticoagulant effect even at very, very, very low doses. So it's one of these ones where we're collaborating with a company in the US about developing this as a therapeutic option for treating this particular type of effect. And work that this company's been doing as well is they've shown that some of the toxic actions that aren't well neutralized, that are acting on the nerves, are also very well neutralized by this medicine. And the reason that it's such a promising target is that this medicine is already approved for human use. Because to bring a medicine from theory to approval, you're looking at about $500 million in cost just to get a drug approved. So if you can repurpose an existing drug, you now have where you've just saved $500 million. And the reason that this is particularly important is that snakebite is a disease of poverty, where it's affecting people the most who can afford it the least. So if it's already approved, then that means their developmental costs are much, much lower in this particular use, which means that you don't have $500 million in costs that you need to recoup by selling this drug to people who might have a yearly income of about $200. So it's a case of where it ticks off every box that you could possibly hammer. So bringing two more examples, bringing it a little closer to home. Australia has the best antivenom in the world. It also has the most expensive antivenom in the world at about $5,000 an ampule. But it's readily available, but does it work against everything? So we looked at the effects of snakes on the blood. Now, things like your tiger snake group, very well known to hit your blood. Things like your red-bellied black snake, very well known to hit your blood. But all the other ones in red were a bit of a surprise. These were ones that we didn't know were acting on your blood. All right, well, how well does the antivenom work? So the ones in red here where the antivenom works, the ones in blue is where it's not really working all that great. The important thing to take home here, though, is that genetic relatedness isn't necessarily a good indicator of whether or not the antivenom is going to work. So within our tiger snake group, great. But within its closest relatives, the swamp snakes, doesn't work very well. Works great against the red-bellied black snake, not as well against the small-eyed snake. So it's kind of scattered across the tree. And that's because venom is such a dynamic trait that's under such extreme selection pressure that means that the organismal relatedness, how closely related species X is against species Y used in the making of the antivenom, is not necessarily going to give you a good predictor. Similarly, we look at the lance-headed pit vipers, where very broad group of snakes extending from Costa Rica down to the tip of South America. These are the snakes that are one of the most medically important groups in the world, huge amounts of morbidity and mortality. So when we tested the antivenom efficacy in blue line, red line is without the antivenom. Against the Sal Bento one, works brilliant, But if we look at some of the ones like Santorim, it works extremely poorly. And this is also our most potent venom. These are snakes that are feeding on amphibians and other animals during the flood season, so there's a high chance of prey escape. So their venom is very different to the snakes that are used in the making of the antivenom. So the ones that are very similar to the ones in the making of the antivenom works great, travel a couple hundred kilometers just within the range of this one snake, still the same species, and you have basically a total failure of the antivenom. Similarly, twig snakes and boomslangs, very similar to each other. Antivenom works brilliantly against the boomslang, total failure against the twig snake, despite them being so close. So the last example is one that really illustrates the problems in the developing world, and that's one of predation by people. Predatory medical practices is something that's a huge issue in the developing world. Saw-scaled vipers occur from Africa up through the Middle East and over and through the Indian subcontinent. The South African Institute for Medical Research makes a great antivenom that it really isn't that expensive for what it is, but Indian antivenom manufacturers are saying, hey, we have saw-scaled vipers, you have saw-scaled vipers, we'll sell you our antivenom for 20% the price of the South African one. But they're selling it without having done any testing. In areas such as Kenya, where they were switching over to the Indian antivenom, deaths climbed 20-fold, which means that everyone who otherwise would have survived is dying. And so the companies are like, not our problem, we'll sell our antivenom to anyone. So we did our testing. We tested snakes from across Africa through the Middle East, established how potent they were, and then we tested the Indian versus the African antivenoms. So ICP and the Samir, these are African antivenoms. ICP is made using the venom of West African snakes. Samir is made using East and West. ICP works great against its target market of West Africa. The Pan-African one works great across. Both Indian antivenoms had a total failure against anything from Africa. An absolute, utter failure. Yet, despite us publishing this 2 years ago and sending these companies a copy of our reports and the Washington Post contacting them when they wrote up about our results, they're still sending their antivenom to Africa. To me, this is murder. If you know that you have a product that doesn't work and you sell it anyway, that's murder. But yet again, this is nothing unique to the developing world where, like I said, these kind of predatory medical practices extend across all sorts of disease states. And I think this is absolutely despicable, and it really, kind of fits with my view of humanity, you might say. Anyway, so thank you very much for your patience, cast of thousands along the way, and I'm sure we have time for some questions. Thank you.
Thank you so much, Bryan. You can see why we got him back, a brilliant storyteller and a brilliant scientist. We are going to take some questions now. If you've got a question there, wave it around in the air and we will come round and collect those. I will load up Twitter and look for questions on here. Of course, next month we're going to take a brief holiday break from BrisScience. So enjoy your Christmas break and catch up on all the science there over your dinner. Make sure you swap some good BrisScience links there. Thank you. Okay, let's take a few questions here. First question is from David on Twitter, who asks, can the cost of drug development be reduced if it is done in the countries that have greatest need? Skip some of the FDA bureaucracy.
Yeah. So yeah, let's just skip all the things to make a drug safe and just go straight and test it in the people in the third world. Yeah, that's never been done before. Yeah, that's actually, that's actually, one of the predatory practices is the testing of drugs in the third world, and that's one of these things that you should be not skipping any steps anywhere. Humanity is humanity no matter where they are. An example of this at its criminal best is during the AIDS epidemic, for example, where the US blood manufacturing companies realized that AIDS was in the blood, so they came up with a way of heat treating their product to kill the virus. Great. What do you think they did with all their stock? They dumped it in the third world. And that's what created the AIDS epidemic in Hong Kong and Vietnam. So, yeah, no, we can't skip any steps for any drug development at any level.
Well said. Howard would like to know, are snakes poisonous to themselves?
It's a very good question. We'll move aside the semantics of poisonous versus venomous. Typically, they aren't sensitive to their own venom, because otherwise, if a cobra bites its lip, it's going to die. So they usually have modifications either in their blood, like antibodies to mop up the venom components, or they have physical changes to their nerves in such a way that their own venom can't bind to its own nerve.
Philip would like to know, what is the evolutionary basis for the selection of red for danger and not some other colour? And perhaps I'd maybe extend that more generally. Why colours or bands? Why do you think that is such a typical indicator?
Well, it's a good question. So one of it is that red is an uncommon color in nature. Like if you're selecting, like green wouldn't make a very good warning coloring because there's so much green already out in the world.
Trees.
Of course, if something's colorblind, it's not going to work very well. But a lot of animals can actually see color. And as far as banding, that means that it's conspicuous even to colorblind animals. So that sharp contrast is really going to make the animal stand out.
I've got a question here. Did you learn anything from your jellyfish studies other than the liquid snot?
Yeah, well, we did develop a new way of milking jellyfish where, like, when they talk about jellyfish first aid, they talk about pour vinegar on because that stops the jellyfish tentacles from firing more. They say never use ethanol because that causes the jellyfish tentacles to fire. So we went, great, we'll use ethanol. So we started dipping jellyfish tentacles into ethanol, causing all the nematocysts to fire at once, and that's how we were able to get our jellyfish venom.
There you go, more Christmas dinner conversation. We have time for just a couple more questions. Someone who asked, Roald Dahl said the black mamba snake attacked without provocation. Is this really true?
No. It's one of those ones where, of course, there's, snakes have more myths about them than anything else. Like if you were bit by a snake and you die in Africa, of course it was a mamba. If you survived, of course it was something else. So some snakes are very, very defensive. And so when you have something like a mamba, this is an animal that hunts using its eyesight, much like a taipan. So they're an animal where they can spot a mouse moving 7-10 meters away. So that means that by the time you're within a couple of meters of it, you're way inside of their threat bubble, and they're really agitated long before you even see it. They've seen you, and they think that you're a predator steadily walking towards them. So that's why they appear to bite without provocation, but they've been clocking you for the last 15 meters.
Fantastic. All right, we are almost out of time. We have one final question, which I'd like. You mentioned your favourite snake. What is your least favourite snake?
Well, I don't really have a least favorite snake. I just have a least favorite snake experience, and that was the one that bit me, ruined my blood. First off, it started off as my favorite snake experience, where the first major symptom was bleeding out of my eyes, which is the most metal thing I've ever seen. I wish I had a picture of it. It was really, really cool. But then life took a very uncool turn, and that's when I bled out of my butt for 18 hours. And it doesn't matter what it takes in life by the time to get to anal bleeding. By definition, it's not cool.
Well, I can't think of a better way to end the BrisScience series for 2019. Bryan Fry, everybody. Give a huge round of applause.
Thank you.
We'll be back in 2019. Head to our website, send the mailing list. Thank you very much. See you next year. Merry Christmas.